My name is Drew Bridges, and I'm a postdoc in the Bachelor Lab at Princeton. And today I'm going to talk about Vibrio cholera biofilm dispersal. Bacteria frequently form surface-associated communities called biofilms. And biofilms are formed by a self-secreted extracellular matrix that allows the bacteria within to collectively resist threats. And these include antibiotic resistance, where biofilm cells are often more resistant to antibiotic treatment than their free-swimming counterparts. Biofilms are also resistant to predation by phagocytic cells, whereby the large size of the biofilm makes them impossible to engulf. And finally, the surface-associated characteristic of the biofilm makes them difficult to dislocate due to flow, for example, in a catheter or in a pipe. So biofilms are highly relevant in many industries and also in the clinic. The biofilm life cycle occurs in a developmental process that's characterized by three distinct steps. First, when a free-swimming cell encounters conditions that favor biofilm formation, it attaches to the substrate. Next, through repeated rounds of cell division and continued matrix secretion, the biofilm matures. And finally, when conditions are no longer favorable, the biofilm disassembles through a process called biofilm dispersal, and the liberated planktonic cells are free to stake out new territories and to repeat the life cycle. In general, in many bacteria, the attachment and maturation phases of the life cycle have been very well studied, whereas dispersal, which is my current area of interest, is very poorly understood. So in Vibrio cholera, the organism that I study, and the cause of the diarrheal disease cholera, the biofilm life cycle is intimately connected to the disease process. Vibrio cholera forms biofilms on chitinous surfaces in small animals and coastal seawaters. The disease starts when a person drinks the contaminated water, and upon ingestion, the biofilms disperse, and liberated planktonic cells are highly virulent and they colonize the small intestine. Notably, cells that are liberated from biofilms are much more virulent than their planktonic counterparts, and even crude filtration of these large particles really reduces cholera transmission. And once the infection has been established, the patient experiences diarrhea, which leads to release of Vibrio cholera, and in many cases returning them to the water supply where the process is free to repeat itself. Thus, we think it's really critical to understand all stages of the biofilm life cycle, especially biofilm dispersal, as it's intimately connected to the disease process. And before I show you my work on biofilm dispersal, I want to talk briefly about what we know about Vibrio cholera biofilm formation. Because of the relevance to disease and its tractability, Vibrio cholera has become a robust model organism for biofilm formation. Traditionally, the assays used for Vibrio cholera biofilm formation have included measuring the amount of biofilm formed on the side of test tubes. As you can see in these test tubes, some mutants exhibit increased biofilm formation, whereas others display less biofilm. Another traditional metric for biofilm formation is colony morphology on agar plates. As you can see, this colony is smooth, exhibiting little biofilm formation, while wrinkled or rugose colonies are characteristic of a strain that produces abundant extracellular matrix. So unfortunately, these assays don't tell us anything about biofilm dispersal, but they have revealed the components that are required for biofilm formation. And from these types of studies that have taken place in numerous labs, we now know that the biofilm matrix is composed of extracellular polysaccharides, shown in gray, and proteins, shown in red. The polysaccharides act like a netting that holds cells in place, and the proteins facilitate cell surface attachments between the cells in the polysaccharide matrix, and cell-cell attachments. Together, these components make up the extracellular matrix that gives the biofilm its tough characteristics. More recently, newer approaches for studying biofilm formation, pioneered primarily in my postdoc advisor, Bonnie Bassler's lab, have made it possible to determine biofilm architecture and dynamics at the single cell level. Using confocal microscopy, it was shown that bacteria within biofilms organize into a reproducible architecture. And just a few months ago, we published a paper in which we used light sheet microscopy to image the dynamic process of biofilm formation from the founding cell to the development of a large community. And you can see in these chemographs the trajectory of individual cells over the course of biofilm formation. And while these approaches have been incredibly informative, we've not yet been able to use these tools to study the biofilm dispersal phase of the life cycle. So what I hope you've taken away from this introduction is that we now know a good deal about how biofilms form. However, we don't know much about how biofilms go from a mature biofilm to biofilm dispersal, where cells are released. And when I joined the Vassar Lab, I wanted to study this biofilm dispersal process, and my goal was to uncover the molecular mechanisms that control biofilm dispersal. But I had to overcome the problem that existing assays do not address biofilm dispersal. Thus, my first task was to develop an assay. So I was able to iterate until I found conditions where when I inoculated cells at very low cell densities and performed bright field microscopy, I could follow the entire biofilm life cycle from start to finish. I could easily observe the attachment and maturation phases, and after about 8 hours, the biofilms completely dispersed. And then subsequently, I switched to using lower magnification imaging so that I could observe many biofilm formation and dispersal events in a single field of view, as you can see here. And what I can then do is to segment these images and measure biofilm biomass within each field of view over time. So I'm going to show you many of these types of graphs today, and in these plots, time is on the x-axis, and the y-axis represents the measured biofilm biomass. And as you can see, I found that for the wild-type strain, biofilm biomass peaks around 8 or 9 hours, and dispersal is completed by around 12 hours. So great, now I had an assay in hand that allowed me to study dispersal. Now what I wanted to do is to identify the molecular components that are responsible for carrying out biofilm dispersal. Given that the wild type grows into biofilms and subsequently disperses, we reasoned that we could use this assay to identify mutants that fail to disperse. The plan was as follows. I would generate an arrayed library of mutants in 96-well plate using colony picking robotics. I then performed high content microscopy on the 96-well plates at 9 hours to ensure that a biofilm had formed, and again at 14 hours to determine if the biofilms had dispersed. And then finally, I identified the hits that retained biofilm formation at 14 hours, and then using PCR sequencing, I identified the gene that was disrupted, and I subsequently followed up on the results. So this is what the screen looked like. Here's an image of a 96-well plate at 14 hours, and you can see only 2 of the wells have biofilms remaining at the 14-hour time point. So this would be an example of a hit, or 2 hits. I then screened around 7,000 colonies in total, and I found 10 unique hits that displayed dispersal defects. So here's a movie that summarizes the dispersal defects for the 10 mutants that I identified. You can see wild type here in the top left corner, and as I've shown you already, it forms biofilms and then it disperses. And as you look around at these mutants that fail to properly disperse, what you'll notice is that some of the hits have really extreme dispersal defects compared to wild type. For example, DBFS here, which is completely deficient in biofilm dispersal. Other hits possess relatively minor dispersal defects, such as BIP-A, which you see here. It mostly disperses, but a small amount of the biofilms remain at the final time point. This shows that our screen was robust, given that we were able to detect such a range of dispersal defects. And we found that the hits from the screen fell into one of 3 categories. The majority of the hits, 7 out of 10, shown in blue here, were hits involving signal transduction. 2 of the hits were in matrix digestion enzymes in green, and 1 hit in red involved cell motility. Today, I want to talk about our follow-up studies on select hits from each category, and I want to mention that we think these 3 categories represent the chronological steps that occur during biofilm dispersal. First, the signaling events occur that permit dispersal. This then leads to the activation of matrix digestion components, and then finally cell motility kicks in, and this allows cells to swim out of the biofilm. So I'll start where I believe that the dispersal process starts, and that's with signal transduction. So the signaling hit that I'll discuss today was in a never before studied 2-component histidine kinase that we decided to name DBFS for dispersal from biofilm sensor. As you can see again, at 16 hours for the wild type, the biofilms have dispersed, whereas in this DBFS mutant, there was no dispersal. And when we quantify this, we see that while again the wild type biofilms grow to 8 or 9 hours and then disperse, the DBFS mutant displays an extreme dispersal defect, and it essentially never appreciably disperses from biofilms over the course of the entire assay. So DBFS, like other bacterial two-component histidine kinases, contains a periplasmic sensor domain between the inner and outer bacterial membranes. It contains a transmembrane domain that spans the inner membrane and a kinase/phosphatase domain. So these proteins typically switch between kinase and phosphatase activities dependent on the ligand occupancy of the sensory domain. This family of proteins is really highly conserved in bacteria and is largely how bacteria respond to external cues. So DBFS is encoded right next to a response regulator that we assume DBFS must signal through. And so we named this response regulator DBFR for dispersal from biofilm regulator. Based on the knowledge of other two-component systems, we presume that DBFS must phosphorylate or dephosphorylate DBFR dependent on its ligand occupancy. Typically, the phosphorylated version of the response regulator binds to DNA. This alters transcription, yielding some sort of phenotypic change. And in this case, we would predict biofilm dispersal. So we'd wanted to test this model. So again, when we get rid of the receptor, the biofilms stay put. And we suspected that in the absence of DBFS, DBFR must be dephosphorylated since we took away the DBFS kinase. And since we predict that the phosphorylated DBFR is the active version, we predicted that deleting DBFR should look the same as deleting DBFS, since either way, the phosphorylated active version of DBFR is eliminated. So we made the deletion and did the experiment. And to our surprise, deleting DBFR did not yield a dispersal defect. Instead, the DBFR mutant dispersed just like wild type. So initially, we were really perplexed by this result, and it was time to go back to the drawing board. So we got to thinking, and we reasoned that perhaps this signaling circuit is not like conventional two-component systems. Perhaps DBFR is not actually the partner response regulator to DBFS. Alternatively, DBFS could signal through some other response regulator that's not DBFR, and the phosphorylated version of this response regulator could activate dispersal. And there's a relatively easy way to test whether or not DBFR is the partner response regulator to DBFS, and that's just to construct the DBFS-DBFR double mutant. If the double mutant fails to disperse, just like the single DBFS mutant, then we would predict that perhaps indeed DBFS is signaling through some other response regulator. However, if the double mutant looks like the DBFR single mutant, then we would know that DBFS does indeed signal through DBFR. And indeed, what we found is that the double mutant disperses. And so, in fact, DBFS does signal through DBFR. Again, if DBFS signaled through a different response regulator, we would have expected to see that deleting DBFS and DBFR together would have yielded a strong dispersal defect similar to the DBFS mutant. So what's going on here? First, what I've shown you is that DBFS is required for dispersal because, as we've discussed, the DBFS mutant has this really strong dispersal defect. Second, I just showed you that DBFS does signal through its cognate response regulator, DBFR, and we know this because the double mutant phenotype shows that DBFR is epistatic to DBFS. And finally, something that we've not yet discussed is that DBFR does not appear to drive dispersal. And we came to this conclusion because the DBFR mutant disperses just like wild type. However, what we now think is that it could inhibit biofilm dispersal. So with this information in mind, we could redraw our pathway. We came up with two potential models that could explain our observations. One possibility is that the dephosphorylated version of DBFR drives dispersal. In this case, deleting DBFS will lead to accumulation of dephosphorylated DBFR, and biofilms would stay put, explaining the dispersal defect for the DBFS mutant. Deleting DBFR or DBFR and DBFS together would allow normal dispersal to take place, consistent with our model. We'll call this model 1. Alternatively, the phosphorylated version of DBFR could inhibit biofilm dispersal. If this were the case, we would predict that DBFS would function primarily as a phosphatase under the conditions of our experiment, and some other unknown protein would be the DBFR kinase. In this case, when we deleted DBFS, we would remove the phosphatase activity, leading to DBFR phosphorylation, and biofilms would stay put. When we delete DBFR, or both DBFR and DBFS, biofilms would disperse like wild type, which is consistent with our data. So we'll call this model 2. So how do we distinguish between these two models? The bottom line is that we needed to determine whether the dephosphorylated or the phosphorylated version of DBFR inhibits dispersal. And the easiest way to do this is to generate a non-phosphorylatable allele of DBFR, and so that's what I did. If the non-phosphorylatable allele inhibits dispersal, then this would favor model 1, in which dephosphorylated DBFR is inhibitory to dispersal. For model 2 to be correct, the phospho-dead allele would not inhibit dispersal, and instead the strain would disperse just like wild type. So of course I did the experiment, and again wild type, as I've shown you already, grows and disperses, and what I found is that the DBFR phospho-dead mutant disperses just like wild type. And furthermore, even when I delete DBFS in combination with this DBFR phospho-dead allele, biofilms disperse just like wild type. And this is because DBFS is unable to phosphorylate this phospho-dead version of DBFR. So this favors model 2, and we can infer from these results that phosphorylated DBFR inhibits dispersal. Again, in this model, DBFS functions as a phosphatase in the conditions of our assay, and our results demand that there's another kinase, and if so, it makes a prediction. For wild type, we would expect to find that DBFR is largely dephosphorylated because of the phosphatase activity of DBFS. This allows biofilms to disperse, as occurs in wild type. In contrast, we would expect that when DBFS is deleted, we see high levels of, of DBFR phosphorylation, and that would explain the dispersal inhibition that we see for this mutant. And then what we wanted to do is measure DBFR phosphorylation in vivo. And to do this, we used PhosTag gels, in which phosphorylated species migrate slower than the dephosphorylated version. And indeed, what I observed was that in the wild type, DBFR is largely dephosphorylated, whereas in the absence of DBFS, we see highly phosphorylated DBFR. And this is really exciting to us because it confirms our model that some other protein contributes to dispersal inhibition by acting as a DBFR kinase. And while we don't yet know the identity of this kinase, it's something that I'm actively working on. So now we have an understanding of how the signaling circuit works, but what I haven't told you is what phospho-DBFR regulates downstream to inhibit dispersal. We reasoned that one possibility was that it inhibits genes that facilitate biofilm dispersal, such as matrix degradation enzymes. Another possibility is that it activates matrix biosynthesis, and as a result prevents biofilm dispersal. At the time, we didn't know what the dispersal genes were yet, so we started by examining matrix biosynthesis first. To do this, we simply used a reporter for gene expression from the Vibrio polysaccharide operon. This is simply a fusion of the VPSL promoter to luciferase. On the y-axis here is the reporter output in log scale, and on the x-axis is cell density. So for wild type, we see that matrix expression is high at low cell densities and turns off as cell density increases. And this makes sense for the biofilm life cycle that I've shown you. What we then found is that in the DBFS mutant, matrix expression is increased tenfold throughout growth. So for now, we are able to say that this pathway regulates extracellular matrix production, and this makes sense that if you're producing a ton of biofilm matrix, it's going to be impossible for biofilms to disperse. And we're currently using RNA-seq to identify the entire DBFS-DBFR operon, regulon, and we're particularly interested to see if the matrix digestion enzymes are regulated by this pathway. So there are many future directions that I'm currently working on with this pathway. I'm currently working to identify the DBFR kinase. As I just mentioned, I'm determining the regulon of this pathway. And the last thing we're really interested about is in determining the identity of a potential DBFS ligand so that we can know what the environmental cues are that this pathway responds to. And we wonder if there are conditions in which DBFS itself can function as a kinase. Okay, so that was one of the new signaling systems that we found controls dispersal. Now I want to switch gears and talk about what we think happens next chronologically in the dispersal process, and that's matrix degradation. So if you're not into signaling, now's the time you can wake back up for the talk. As a reminder, we know that the cholera biofilm matrix is composed of both proteins, shown in red, and polysaccharides, shown in gray. So it was very fitting that the screen revealed only two matrix digesting enzymes, one of which is a known protease, and the other is a predicted polysaccharide lyase that could digest the extracellular polysaccharides. So I'll start by talking about the protease LAPG. Based on substantial previous work, primarily by George O'Toole's lab in Pseudomonads, LAPG is a protease that's found in the periplasm. It's thought to be very specific for digesting adhesin proteins, and these adhesin proteins facilitate attachments in the biofilm. Cutting these adhesins allows the biofilm cells to detach and leave the biofilms, as we see for wild type. And indeed, in the LAPG mutant, which I identified in my screen, which can't cut these adhesins, there's a strong dispersal defect. Furthermore, the previous work showed that LAPG is regulated by sequestration through an intermembrane protein called LAPD. LAPD senses cyclic diGMP, which is a biofilm regulatory second messenger, and when cyclic diGMP levels are high, LAPG is sequestered by LAPD, and this allows for biofilm formation. When cyclic diGMP decreases, LAPD releases LAPG, and it cuts these adhesins, and cells can disperse from biofilms. Thus, if this model were true for our system, we would expect that deleting LAPD would lead to premature release of the LAPG protease, and therefore biofilms would disperse early. And indeed, what we found is that the LAPD mutant exhibited premature biofilm dispersal, as you can see here. Furthermore, we thought that if we constructed the LAPD-LAPG double mutant, because of the lack of the LAPG protease, these should exhibit a dispersal delay just like the single LAPG mutant. And that is indeed what we found, confirming that this pathway is functioning in Vibrio cholera biofilm dispersal. So taken together, this work shows that LAPG is responsible for digesting some of the protein component of the biofilm matrix. However, because these adhesins are only one component of the proteins that make attachments in the biofilms, we thought that other proteases may be responsible for degrading these other protein-mediated attachments, despite the fact that we didn't identify any other proteases in the screen. So to double check, we individually deleted all the known secreted proteases in Vibrio cholera, and indeed, none of these mutants exhibited dispersal defects. And from these results, we believe that LAPG really is the major player in digesting the protein biofilm attachments, but it remains possible that some of these secreted proteases are redundant. So that was the digestion of the matrix proteins by LAPG. And now I want to talk about the potential digestion of matrix polysaccharides by RBMB. So as you can see here, lack of RBMB causes a dispersal defect. And as I just mentioned, RBMB is predicted to digest the extracellular polysaccharide matrix. Like LAPG, the RBMB mutant forms nearly wild-type levels of biofilm. However, it fails to properly disperse. So what we wanted to do is to find a way to determine whether RBMB does indeed digest the matrix polysaccharides. And to do that, we wanted to be able to measure the amount of the polysaccharide remaining at the time point after which wild-type dispersal has occurred. For example, at 16 hours. So remember, in wild type, we have both LAPG to digest the matrix proteins and RBMB, which we think is digesting the matrix polysaccharides. In the LAPG mutant, the adhesin proteins remain because LAPG isn't there to cut them, but RBMB is still there, and so we think polysaccharides should be digested. In contrast, in the RBMB mutant, the adhesin proteins are degraded by LAPG, but we hypothesize that the matrix polysaccharides remain, and this prevents dispersal. So what we did was we measured the polysaccharides in the LAPG and the RBMB mutant at 16 hours, a time point after which the wild type strain has dispersed. Again, both of these mutants don't disperse, and so we think there should be no polysaccharides remaining in the LAPG mutant at 16 hours because of the activity of RBMB. And in fact, when we stain the remaining LAPG biofilms with wheat germ agglutinin, which recognizes the polysaccharides, we see very little signal, suggesting the polysaccharides have been digested by 16 hours. In contrast, we expect to find that biofilms lacking the polysaccharide lyase RBMB should stain strongly with wheat germ agglutinin. And indeed, that's what we found, suggesting that polysaccharides remain at this time point in the RBMB mutant, indicating that RBMB is responsible for digesting the matrix polysaccharides. Furthermore, we can quantify this, and we see that the RBMB strain contains 6 times as much wheat germ agglutinin staining at 16 hours compared to the LAPG mutant. So they're both stuck, but they can't disperse for different reasons. Thus, LAPG, the adhesin protease, and RBMB, the polysaccharide lyase, work together to carry out matrix degradation and therefore biofilm dispersal. Going forward, we'd really like to know how RBMB in particular is transcriptionally regulated and if our signaling system, DBFS, DBFR, that I talked about in the first part of the talk, regulates its expression. Okay, so the signals have been received, the matrix is being digested, and this is where we think cell motility kicks in. And from our hit in the screen involving cell motility, there was only a single hit, and that was in the chemotaxis response regulator KY. So as you can see, the KY mutant is not as deficient at dispersal as some of the other mutants that I've shown you today. However, the dispersal dynamics are significantly different than the wild type. And as I mentioned, KY is involved in chemotaxis, the process by which bacteria move along or against the chemical gradient. KY, it's a response regulator, just like DBFR that I was talking about earlier, and receptors that sense these chemotactic gradients alter KY phosphorylation. Instead of acting as a transcription factor, phosphorylated KY binds to the flagellar motor apparatus, and this leads to changes in the direction of cell motility. So when KY is phosphorylated, cells frequently turn and change directions, whereas when KY is dephosphorylated, or when KY is gone altogether, as in the KY mutant, cells swim straight and are therefore unable to undergo chemotaxis. So as I've illustrated here, the KY mutant is non-chemotactic, and we can measure its turning frequency. So wild type in homogeneous medium turns on average every other second or so, while the KY mutant almost never turns. So we reasoned that the KY dispersal defect that we observed could be due either to chemotaxis failure, or it could be a result of swimming straight. To distinguish between these possibilities, I used a KY phosphomimetic allele that we call KY star. And KY star also fails to chemotax. However, it turns much more frequently than the KY mutant, and it turns much more frequently even than wild type. And we found that for this KY star phosphomimetic mutant, that it disperses just like wild type, telling us that, that changes in direction of swimming motility rather than chemotaxis per se are required for cells to exit the porous biofilm matrix. So our model is that cholera cells have to occasionally reorient to exit the porous matrix as it's being digested. The KY mutant, which only swims straight, becomes jammed and is therefore unable to exit the porous biofilm matrix. The KY star mutant, in contrast, turns frequently, and so it has no problem getting out of the biofilm. Importantly, there's abundance of experimental and theoretical evidence from other groups showing that straight swimming mutants like the KY mutant do not spread through porous media as well as their counterparts that can change directions. And perhaps something closer to home that might illustrate this phenomenon is getting out of a tight parallel parking space. Let's be honest, those of us who drive have all been in this situation. But you can see that this person is reversing again and again and changing directions. They're turning, and eventually, believe it or not, they will get out of the space. They would never have gotten out if they just drove straight, or if they did try that, it would have gotten really ugly. So this is kind of what we think it's like for cells that need to escape the matrix. There are lots of other cells, proteins, and polysaccharides that are being digested, and a cell has to sort of wiggle its way out to get out of the biofilm. See? They actually do get out. So to summarize, today I've told you about how dispersal occurs in Vibrio cholera and the chronological steps that we believe are required. I told you about one signaling circuit that regulates biofilm dispersal in Vibrio cholera, the 3-component system that we've discovered involving DBFS, DBFR, and an unknown kinase. Then I told you about the enzymes LAPG and RBMB that degrade matrix proteins and polysaccharides respectively. And finally, I showed you that reorientations in swimming direction mediated by the chemotaxis machinery are critical for exiting the biofilm matrix. So now in just like 2 more minutes and 3 slides, I want to layer on top of this a brief version of a story about another signaling circuit that controls biofilm dispersal that we published last year. And as I told you, I work in the laboratory of Bonnie Bassler at Princeton. And if you know Bonnie, then you probably know she's a very passionate person, and she's especially passionate about quorum sensing in particular. So it'd be unthinkable for Bonnie to not passionately recommend that I examine the role of quorum sensing in controlling biofilm dispersal. And so it turns out that quorum sensing does also control biofilm dispersal. So very briefly, quorum sensing is the bacterial cell-cell communication process that allows individual cells to coordinate group behaviors. To achieve quorum sensing, bacteria secrete and detect small molecules called autoinducers. At low cell densities, these autoinducers simply diffuse away from cells and individual behaviors are enacted. At high cell densities, when autoinducers have accumulated, the cells detect them and they uniformly enact group behaviors. We found that mutants that are stuck in the low cell density quorum sensing mode are unable to disperse from biofilms, showing us that quorum sensing is another signaling mechanism that controls biofilm dispersal. Vibrio cholera contains 2 well-studied autoinducer receptor pairs. The first involves the autoinducer CAI1, and this autoinducer is produced exclusively by Vibrio cholera and other closely related species. And this autoinducer is thought to convey the density of kin or closely related species in the environment. The second autoinducer is AI2, which is produced by Vibrio cholera, but also many other types of bacteria. And this autoinducer is thought to act as a universal signal for bacterial cell density. Together, these 2 autoinducers, through their receptors, work to control group behavior gene expression in Vibrio cholera, and in this case, biofilm dispersal. So I did a number of mechanistic experiments, and for the sake of time, I'll skip them and share with you our model. We found that endogenously produced autoinducers were detected at different time points in the biofilm life cycle. The kin signal, CI1, was detected when biofilms were very small. In contrast, the universal signal, AI2, was detected much later in biofilm development. We found that both CI1 and AI2 produced by Vibrio cholera had to be present simultaneously to trigger dispersal. And we call this a coincidence detector. And coincidence detectors are present in distantly related cell types such as neurons that use similar mechanisms to process multiple signals. And because of the asymmetric detection of these 2 autoinducers, AI2 is the rate limiting step for the coincidence detector. So I wanted to know that if I added exogenous autoinducers, could I change biofilm dispersal? What I found was that when I added excess CAI1, mimicking a dense but closely related bacterial community, dispersal was not accelerated. And this made sense because the receptor was already saturated and because the self-produced AI2 was still limiting. In contrast, when I added excess AI2, representative of a dense but diverse bacterial community, this prematurely activated the coincidence detector and led to premature biofilm dispersal. And we think this slow detection of endogenously produced AI2 in the face of its own kin signal makes sense because it allows cholera to detect other organisms in its environment. And when these competitors are detected, cholera flees the scene to find a new niche to settle in. And I just want to finish by mentioning 3 of my longer term goals. First, I'm really curious to know how multiple signaling circuits integrate to control biofilm dispersal. For example, how do quorum sensing and this DBFS system that I told you about earlier function together to control the biofilm life cycle? In addition, there are several other signals that we now know about that I didn't have time to discuss today. Next, I plan to study how other organisms influence the Vibrio cholera biofilm life cycle. For example, other bacteria, perhaps through this quorum sensing mechanism that I just mentioned, phages that lead to Vibrio cholera cell lysis, grazing predators that eat bacteria, and potentially even immune cells. If these other organisms alter the biofilm life cycle of cholera, I would love to study the signaling mechanisms that are responsible. Finally, as I mentioned at the start of the talk, We now have tools to image biofilm structure and dynamics with single cell resolution, and now we're coupling my assay with these approaches to study biofilm dispersal at single cell resolution. This approach will tell us exactly how cells leave the biofilm, and I'd love to know if cells leave as a collective, in small groups, or as individuals. Do they leave from one region of the biofilm first, for example, from the outside of the biofilm or from the inside of the biofilm? And with light sheet and single cell resolution imaging, I think we're going to be able to address these kinds of questions. And with that, I'd like to thank the lab, especially Bonnie, who's a wonderful advisor, and also Shinny, who helped with analyzing the swimming reorientations that I discussed in the chemotaxis section. And finally, my funding, especially Damon Runyon and HHMI, who funded my postdoc, as well as the National Institutes of Health. And thanks to Marcus and the rest of the Institute for this invitation. It was it's a really fantastic seminar series, and I'm happy to take any questions.